So now it is my pleasure to introduce our speaker, Ray Simpson. Ray currently works at the Yale Peabody Museum in the Entomology division. His background is in fish symptomatics and taxonomy, but he has a broad interest in insects, mostly Lepidoptera, fish, a Western Atlantic focus, and mollusks. Ray is an active UV lighter and moth collector in the Northeast based out of New Haven. He's going to present on bugs he's observed at Fresh Meadows over the past few months. Take it away, Ray. All yours.
Well, thank you, everybody. Thanks to everyone for coming on a Monday night to this presentation. We had a good time doing this project. Given the COVID situation, is really difficult getting out there and actually doing some socially distanced fun outdoor activities. And this was actually an ideal activity because it was a few of us, we were well spaced out, and we were sampling an area that we've never sampled before. So it was quite a interesting experience, and we found a bunch of interesting stuff. So what exactly, we'll start from the basics. What exactly are we studying? So I'm in the Department of Entomology, and entomology is related to the study of insects, as opposed to other groups like arachnids and crustaceans. Entomology is strictly the study of insects, and it covers everything from the internal anatomy to distribution and the taxonomy or classification of insects to the evolution of insects. Insects themselves are a taxonomic class within the hexapoda, which means 6 legs, distinct from the other major classes of arthropods, which are all considered jointed-legged organisms in the phylum Arthropoda. There are 28 to 30 orders, depending on how you ask, within the insecta in the United States. And actually you can encounter most of them in Connecticut. Some are much rarer than others, but you can. Orders are the larger groups of insects that you hear, like beetles, flies, wasps, and butterflies, are all considered like order level groupings. Bugs, you hear the term bugs, those are technically in entomology circles only considered the order Hemiptera. They're not considered all the insects. Bugs are a type of insect, but not all insects are bugs. So for most of survey work, scientists want to get to at least the family level to be useful for research projects. So if you go down to the family and genus level, as you can see on the right, it shows the Linnaean hierarchy from domain all the way to species. Most scientists want to get their species all the way down to the Latinized genus species name. So in the name, so say the monarch is Danaeus plexippus, you want to have your species identified all the way to that level for it to be useful for scientific study. And a lot of these specimens are IDable by images, but a lot of them do require specimens for exact species identification. So in many instances, we have to be happy with genus or even family for some of our images, just because we don't have the detail needed to ID them. So why do people survey insects? For many reasons. People might ask, why do people collect insects? Why do museums have such large collections? But there's many reasons why it's important. One is preservation, and I like the quote, "You cannot protect what you don't know is there." And that is a really good example of, basically you can't protect anything until you know what species are present. So you don't know anything that's endangered, you don't know anything that's potentially sensitive unless you actually look for them. So when you're setting up protected areas, you have to kind of know what's there. And then there's also the use of historical data, especially with climate change now. There's a lot of species that have actually spread from the south to the north. And in the presentation coming up, I'll show some examples of some of these species that are more southern, that have basically come up the coast. And they basically start at the coast of Connecticut and push inland over time as they adapt over the years to the shifting climate. So species that are found in, say, Virginia are going to start to find coastal Connecticut and New York to their liking. So when we compare this data to specimens in our collections, we could see these changes that happen over time. We could see the range shifts. The third thing is taxonomic. So this is when people are actually describing new species. So this requires taking series of specimens, and as you see in the upper right there, there's a little moth genitalia picture. A lot of times you have to dissect them and study them at the almost microscopic level to be able to identify species. And with the advent of lots of genetic techniques, we're finding a lot of species that were considered one are now actually 2 or 3 or 4 species. So a lot of times when you're doing these type of studies, you need to take multiple specimens. Another thing is environmental health. Insects are a major indicator of ecosystem health. So if you study a particular habitat, you can tell if it's disturbed or healthy based on the insect population there and the really intricate symbiosis between the plants and the animals. So if you have certain species present, it shows basically if the ecosystem is healthy. And then finally, just plain enjoyment. There's a lot of citizen science going on. I'm on iNaturalist. I'm a big iNaturalist fan. And even citizen scientists with very little background in biology can make really big contributions by simply going out there and seeing what's out there. So for this study, what exactly are we looking at? We're actually studying a small plot of land called Fresh Meadows Preserve. It was obtained in 1985 by the Cheshire Land Trust. It's right off Cook Hill Road. It's easy to miss. It's a little parking lot off the left side, and it kind of goes in, and it's only about 33 acres, but it covers a very interesting range of habitats from uplands in kind of the woodland area with hemlocks and pines to aquatic habitats. There's a pond, and then there's also the Mill River that kind of winds through there. So there's a big range of habitats, but the main part that we actually sampled was this large open meadow. You can see an example in the inset of the picture there. There's a large patch of basically open wetland meadow, kind of like a sedge meadow, with a lot of wildflowers, and a lot of potential diversity is found within that habitat. And anytime you have an edge habitat on the edge of a woodland and field, that tends to jack up the diversity because there's a lot more plant diversity in that area. So if you're there, you can kind of see what I'm talking about, but there's actually several trails that go through this property. And it's kind of, you can see it's surrounded basically by residences and businesses, but this open area is in the blue circle you see right there. And you kind of walk in between 2 houses, you're kind of going down a narrow path, and you just come out onto this open field. And it's actually, I think, a gas line right of way. And what they had done there is basically re-vegetate it with typical native wetland vegetation. So they actually did a fairly good job, because when you're there during the late summer, there's a rich diversity of wildflowers present. So what you're seeing here is a typical blacklight mercury vapor light setup. So we set up on 4 separate nights during this, basically the summer and early fall. And then Steve went twice during the day. So we did 1 in July, 1 in August, 1 in September, and 1 October. And we stayed from basically like nightfall, like whenever dusk was. You can see how it gets later or gets earlier as the fall progresses. But you stayed till around 12, 1, 2, depending on the conditions and the amount of moths and other insects we were seeing. Steve went during the day and took photos of things that were on the flowers by day, but this is more, I was present for only the nocturnal surveys. What we use for equipment were mercury vapor lights, which are the really bright lights, the sun lamp type lights. They're 250 and 175 watt. And then we had the blue lights, which are the 15 watt black lights, and at least 2 sheet setups. And sometimes we did 3 sheet setups, depending on how good of a night it was, how good we felt. And the nights were chosen specifically to maximize what we could find. So we kind of want a warm, humid weather with low moon, like dark, and no wind, because I find that wind is probably the worst for lighting. And this is actually a sheet from the top of Sleeping Giant, which is fairly close by, but it's a good example of what could happen when you have a really good night. And this is one of our really good sheets from the late summer up at the top of Sleeping Giant. So a little bit more, these are more views of the lights and setups. So the mercury vapor is on one side and the black light is on the other. We use the mercury vapor to bring in stuff from a distance, so they have a radius of basically like the best activity is restricted to a certain radius. So the bright one will be able to draw moths and other insects from further away. And then the black light is really helpful in drawing things that are in the periphery of the sheet, drawing them into the sheet. So it's often good to use two separate lights at the same time on the same sheet. And we just kind of just go over there and document what we found. Steve has a DSLR. I have just my phone, and I take pictures with my phone pretty well on the sheet. And the diversity can be quite epic on some nights during the summer. So a lot of people ask this question. This is kind of a good basic question of why do moths get attracted to light? And it is a thing that's still a little mysterious, but it has to have something to do with the navigation that the insects use with the stars and the moon specifically, because the moon is the brightest light in the sky. And the moonlight, because of how far the moonlight comes in, is their parallel rays. So the moths could actually use the parallel rays of the moon to navigate by. And since the moths will never be able to get to the moon, they never pass it, it's almost a fixed beacon in the sky that will allow them to navigate. But when you replace the moon with a bright light source like a lamp, the moths are able to basically approach it and circle it. So they become confused because they're seeing this normally parallel light source is now coming from all different directions. And so what happens is the moths tend to spiral. So you'll see this when they're coming into the sheet, they'll kind of do these spiral, cone-shaped spirals and come into the light and pass it and crash into the sheet and then fly off again and then do the spirals again. So it definitely has something to do with messing up their kind of internal guidances. And the moths will be very, they'll only do this a couple times and they'll usually end up just like this picture here. The moths will usually end up resting on the sheet. And a lot of them will stay the entire night just sitting there. They will not flee or fly off or beat themselves up. Most of the moths stay in very good condition through the course of the night because they tend to rest on the sheet by the time they realize that the light source isn't helping them navigate anywhere useful. So here, here's an example of our Naturalist page. And our Naturalist page is, if people haven't used this before, it's basically submit records that you find based on pictures. So you take a picture and then submit it and then record data as in when it was taken, what time and what location. And people will come in and back up your ID. So if I say that one, I ID'd that one as a Lafria versicolor, that was my ID. It doesn't have any sort of significance to it yet. But if you see on the bottom, there's that jumping bush cricket. The jumping bush cricket's at research grade, which means somebody has come in from outside me, another person has come in and verified my ID is correct. So by doing this, you can basically assemble life lists. And as you can see, you get 340 observations, and then it sorts it by the number of species. So it's really useful to keep lists of species that you've seen on any particular trip. Like here, I've actually searched only Cheshire. So I'm only looking for the species that I've seen in Cheshire, which is fresh meadows. So I highly recommend using it. It takes a little getting used to, but once you get to use it, it's really fun. Within fresh meadows itself, the plant diversity is quite mixed. I have some examples here of some of the plants that you'll see if you are there from July to October. There's a mix of exotics, like there's these amur maples, which are actually from Asia. There's several plants that grow on the property, and they're actually one of the major maples that are growing adjacent to these meadows. Purple loosestrife is another one a lot of people are probably familiar with. And then there's some natives like bayberry, which is actually not the commonest plant on the coastal plain. It usually occurs in poor soils or even near the ocean, in salty soils. So this is kind of an interesting plant to find. And then various fern species, which are usually ubiquitous in eastern woodlands. as far as the plants that are of interest to a lot of our pollinators and a lot of our native butterflies and moths, there's 4 main groups that we saw in the meadow. These are the eupatorium, the Joe Pye weeds, ironweed, which was very common in the meadow, solidago and other genera of goldenrods, and then various asters, which are super common as you got into the fall. And they're usually those little white flowers. So what did we find? So I'm kind of gonna break it down to stuff of note that we found, and then kind of a slideshow of the interesting species that we've encountered during the course of the summer. So according to my data, I have 339 observations of 233 species. Danny has 65 uploads with 35 species, and Steve has 242 of 193 species. Some overlap there, like there's going to be some that are doubled, but for 4 trips at night, that's a pretty significant amount of diversity for this small patch, surrounded by houses, basically. So keep in mind that this list is also provisional. Like Steve has a lot of material that he's still identifying and adding as the winter goes on. And there's a lot of stuff on iNaturalist that's not even included as a species until you get it to the species level. So there's a lot of ones that are represented just by family or even order. So that number will only increase over time as people vet these identifications. The list here, Steve was kind enough to send a list of some of these notable records that we have encountered over the fall. And a lot of these are based on iNaturalist, so I wouldn't take these seriously as in their state records for science, because they're probably just state iNaturalist records. But a lot of them are quite rare moths, and since moths are very well documented on iNaturalist, if you have any state firsts, that's pretty good. And a lot of these, as you can see, are very small. Like all the species that you see there except for number 8 are probably under a half an inch in size, and a lot of them are only a few millimeters in length, like 5 millimeters. So a lot of these are what you'd call micromoths, and micromoths are one of those groups that are very poorly studied. And if you take the time to actually take photos of these tiny moths, you have a really good chance of finding species that are very poorly known from the area. So you can see a lot of them. There's a state first for a lot of these species, and a lot of these species don't have any other records on the map for iNaturalist, so they're really impressive. That Coristonura houstonana is particularly cool because there's only a few records that were found in the entire New England area for that particular species. As for cool, like more charismatic big stuff, the Fresh Meadows Preserve has a huge population of Chinese mantids. Like there are probably hundreds of them in the meadow. When we went there in July, you can see there are nymphs, they're babies or young. And then when we went there in August and September, they're fully winged out adults. So the females and the males, when they're fully adult, will have well-developed wings. And the nymphs, like on the far left, are wingless and they're smaller. So they grow fairly fast during the course of the summer, and they are very ravenous. So the Chinese mantid is an exotic species. So contrary to a lot of people think, like it's not illegal to kill mantids, and it's probably in certain situations probably better to thin out their populations a little bit, because they're very voracious predators of lots of native pollinators. You can see they even eat stink bugs. That one in the middle that's eating a brown marmorated stink bug, which a lot of people are familiar with as a pest. So these mantids are not very picky, and they eat a lot. So you can imagine just how many insects that these hundreds of mantids in this meadow eat. And when we went in October, we actually could see the Uthika. So that bottom right photo actually shows the egg mass of a Chinese mantis. And the adults pretty much died off. By October, all the adults were dead. So they have a fairly fast life from being young in June and then dying off in October. Speaking of mantis, the Connecticut state insect is Mantis religiosa, which is the European mantis, which is kind of strange because the European mantis is an exotic species, so I don't know why it's Connecticut state insect. But it is noticeably rarer than the Chinese mantis. Like in our time that we spent at the Fresh Meadows, this was the only individual of the Connecticut state insect that we actually found. There's a heck of a lot more Chinese mantids than there are European mantids. And it's a bit disturbing because the religiosa is a lot smaller. So there's probably a lot of, I wouldn't say it's not cannibalism, but predation going on this mantis by the other mantid species that are there, which are much bigger. The Chinese mantids are probably 1.5 times the size of these guys. Another cool thing we saw, which is basically like a mini, it's like a mini mantis, as you can see from the middle photo, is they're small neuropterans related to lacewings, and they're called mantid flies. And the cool thing about it, it looks like a lacewing with a lacewing butt and a lacewing front, or a mantid front. So they have little raptorial legs, just like a mantis, but the back part is more like a lacewing. And their predation style is very much the same. They're ambush predators, and they strike out against anything in their size range on the sheet. Even things up to like 1/3 of their size, they'll actually attack on the sheet. So they're pretty bold. So the one in the middle has aspirations to be a female mantis, but they're pretty good. They're pretty ferocious for their size. There's 2 main species, is the brown and the green. And this year was a particularly good year for them. We're seeing them into September. One of the cool things, and I have had a lot of, I'm a moth person, I'm a moth fanatic, but one of the coolest finds that we found during the course of this field season was this Papapima, the ironweed borer. And I actually had a couple back and forths on email about these with the land trust, and I had predicted that we'd find them based on the amount of ironweed there. And then we went there a few weeks later and ran into them in large numbers. And we actually saw 17 in September and 3 in October, and that's impressive because the Papapima sarasata is a state-listed species. And so it's good to see numbers like that. And like a lot of these Papapimas, they tend to be rare and localized, found within maybe a few hundred yards of their host plant. If you were at one of those houses that were in the vicinity of the land trust, you probably would never see these at your porch light because they're so limited to where their host plant is found. So this is a really cool find. Another really, like this is pretty good compared to even the Papapima, is this Dnuma deponens, which is an exotic species, but it's the first Connecticut state record ever. It was only first discovered in Georgia in 2012. So you can see on the map, it's spread pretty significantly west and also northeast. And it's probably following the coast and riding up the coast in the proper climate zone. And it has spread pretty well, and I think the first records were also found in New York and Massachusetts this year, so they have migrated up the coast in fairly strong numbers. And it's a fairly decent sized moth. It's over 1 inch in length, and it's very beautiful. If you look at it at the correct angle, it has this kind of blue-green sheen to it that you can't see in this photo, but it's a very interesting and pretty moth. But unfortunately, it's an exotic. And if people are familiar with the fringe tree or the silk tree, that's albizia. Those are widely planted, naturalized in Connecticut, and that's the host for this particular species. So it's likely we're not going to see the end of these in the future. So here I'm going to kind of show a slideshow of some of the interesting moths and other insects that we found over the course of the summer. One of my favorite groups is the catocala, which are called the underwing moths. The catocalas are usually distinguished by camouflage forewings and very brightly patterned hindwings. So Catocala ilia and Saragama are very distinct species. One has this burnt orange, pumpkin orange colored hindwing, and one has this beautifully yellow banded hindwing. There's another Catoccolum ulercula, which actually is only found on bayberry, which is that plant I showed at the beginning of the presentation that's kind of localized. So seeing Catoccolum ulercula is that's a fairly good species. That's probably the best Catoccola species we saw there this season. We saw additional Papapima, including Impecanosa, which is a aster feeder, and Baptisiae, which is actually, it feeds on Baptisi, which is like wild indigo. I didn't notice any wild indigo there, but it's obviously present somewhere in that meadow. Ativa aurea is a really pretty moth, but it's actually another species that's found on an exotic host, which is Ailanthus, which a lot of people in cities are familiar with Ailanthus trees. And Rhosedra on the right there is a large noctuid that is also introduced, I think, from Europe. And it's fairly recently established in Connecticut, and I found it everywhere from Canaan in the Northwest all the way to the coast. So it's present in all of Connecticut now. Some other pretty moths that I decided to include here, including emeralds, sphinx. We only had a few sphinxes because we were kind of late in the season for sphinxes, but we did get a Dorapsomyron. And there's some really interesting micro moths that are also present, slug moths, tiger moths, and that Perigeus anthoides is another ironweed feeder, which I found out later is kind of an interesting record because it's one of those species that is migrating up the coast. And it's one of the few records for this part of this, of New England for this particular species. Beetles. We had a decent diversity of beetles, including lots of weevils, lots of small beetles, like minute beetles that we're still in the process of IDing. But here's some examples of some of the interesting diversity of beetles that we saw. The ball on the right is from our first night there, and the first bugs that showed up in all of our sampling were these Exomala orientalis, and they came in such big numbers, they formed these big mating balls. And basically they're in the passion, I don't know. And they actually were so embroiled in it that they actually tried mating with the Cyclocephala, which you can see on the bottom left. There's two different types of beetles trying to breed with each other, which probably won't turn out too well. The Lebia is a really pretty green beetle. Copris is a type of dung beetle, so we had both males and females in October, which is quite late. Orthosoma is a large longhorn beetle, a very big, probably about like over 2 inches in length, impressive longhorn beetle. There was a lot of Asian ladybugs, to keep with the theme of exotics. There was a lot of Asian Harmonia ladybug beetles, which basically coated the sheet from usually along the top by the end of the night. There was a great diversity of pollinators, and with Steve's pictures that he took during the day, you're able to see the diversity. Most of the daytime pollinating was done by Hymenoptera, so you can see various types of wasps there, from the Circeris, the ones that gather beetles and put them in burrows. And then you have that's a chrysidid, a cuckoo wasp. And then you have butterflies, which are the main, like the more conspicuous pollinator. But Hymenoptera and butterflies are the dominant daytime pollinators. But at night, on the same goldenrod patches, I saw a lot of moths. So on the left, you see the two pictures of nighttime moth pollination, which is probably underestimated and probably represents a significant amount of pollinators in addition to Hymenoptera and beetles, which are probably just as important. Here's some examples of other various types of insects that we saw. Katydids and grasshoppers are quite common in the meadow, not as common at night, but they're quite common during the day. Katydids are the ones that you hear chirping at night alongside the crickets. And they usually have the long antenna, and they're usually green or brown in color. The grasshoppers don't normally make sound unless they're flying. But most of the grasshoppers are very important meadow associates. So grasshoppers are very important indicator species of meadow health. So seeing a lot of grasshoppers is pretty good for showing the health of a meadow. We had various assassin bugs, spiders. That wasp is an ichneumon, which is actually a parasite. There's actually a very high diversity of parasitoid wasps, which kind of is an indicator of the diversity of the habitat. A lot of these ichneumon wasps are basically parasitize other insects, from caterpillars of Lepidoptera to grubs and larvae of other types of insects as well. And there's all shapes and sizes of ichneumon wasps on the sheets at night, and probably just as many in the fields during the day. As far as other, as I mentioned, the chirping Orthoptera that were found at night, which are katydids and crickets. There's these big angle-wing katydids, and then there's the smaller crickets that are living on the vegetation and chirping at night. So tree crickets and bush crickets. And then I think this is my final picture here. These are like various Diptera and caddisflies and other miscellaneous stuff that we saw. The crane flies are fairly diverse, lots of different types throughout the season. Probably the most diverse and underappreciated group is the plant hoppers. And I know Steve has a ton of pictures of plant hoppers that he's taken over the course of the summer. So many different colors and varieties of plant hoppers, and they're very small, so they're hard to appreciate, but they're very important for meadow health as well. And then you had caddisflies, which is an indicator of the meadow, the wet meadow and the Mill River, which is where most caddisfly larvae live, is in the streams and ponds. So in conclusion, there's still a lot more we could do. We only covered half the flight season. So April, May, and June are kind of the peak for Lepidoptera diversity in Connecticut, and we didn't even get there. The earliest we were there was early July. So we missed a lot of the big moths like the Saturniids and Sphinxes, and a lot of the first brood of a lot of the species that are found in Connecticut that come out in the spring, and then they come out later in the summer. Of course, we did only lighting. So if we tried different sampling techniques and utilize traps, we would probably increase the species list, but a lot of these would involve collecting, so not sure that's allowed on the property. Daylight surveys would add more butterflies. Steve only reported 3 butterflies and 2 skippers, but there's definitely more. There might be over 24 species of butterflies that are found in that field by the end of if you did a full survey. The biodiversity is quite high. There's a lot of species diversity. It's not just a lot of one species. A lot of polluted and degraded habitats have a few species. If you have a survey where you have a lot of diversity, that's an indicator that the habitat is pretty healthy and there's a lot of plant diversity. So that's a really good sign. And if we tried different habitat types, like maybe the upland, the wooded area, or the swampy bog area, or along the river, we'd get lots of different species that we didn't get in the field. So we would definitely be in favor of continuing in the future, COVID willing. So I'd like to thank my colleagues at the Peabody, Danny Brass and Steve King, for helping with the surveys, and the Yale Peabody Museum for providing equipment, and thank you for everybody on iNaturalist who helps with the identifications, because that really helps us a lot. So I'm up for any questions.
Ray, first, I just want to say, wow, 339 observations of 233 species. That's impressive. What a great activity to do during COVID. That's so fun. At least you had something great, a passion for something that you can do, because you know, we're all looking for that, you know. And I love that. Yep.
Oh, sorry, you're gonna finish?
No, I was just gonna say, and it's nice to know we have indications of so many indications of a healthy meadow.
I had come in thinking, just from first impressions, I was thinking like, okay, we're surrounded by a neighborhood and we're in New Haven County. Usually I underappreciate the diversity of New Haven County, given that there's a lot of urban around here. So I came in with lower expectations, but came out of this with a bigger appreciation of these small patches of habitat within the urban and suburban environment. These are essential for preserving biodiversity and giving these species refugia from disturbance. And I'm glad that these spaces exist.
Yeah, awesome. So we have a couple of comments, mostly in the chat box. Let's see. Karen, I don't know if you want to unmute. She's asking the best way to kill these mantids. I'm not sure which ones you're referring to. Should we dispose of the Chinese mantis egg cases? That's the question there. And Karen, feel free to unmute so you can have a conversation with Ray.
So where are we? Yeah, I just de-screen shared myself. So
thank you. Yes, we can see each other. Everybody, feel free to share your screen so we can see you.
So I'm going down. Best way. Okay, now I'm reading it. What is the best way to kill the mantids? What should we dispose of the Chinese? I never like the mantids. To be honest with you, mantids are extremely intelligent insects, and they're like they look at you in the eye. They're charismatic, and I'm never a fan of killing things like killing insects like these. But they are an exotic species, and at the densities that I'm noticing in the meadow, I think they're actually becoming probably detrimental to the pollinators in that field. And they do eat monarchs. They eat pretty much everything. And by the end of the season, it's like, who knows how many thousands, tens of thousands of insects they take. So it's a lot of times people will, to dispose of the mantids, it's like you can, you would have to self-harvest them because of the fact that they're so large. You could kill them as you find them. You could step on them or do something like that. But I'm like, it doesn't sit right with me to tell people to just go around stepping on mantids. Yeah. But a more humane way to kill them is to put them in a freezer, gather them in a container and put them in the freezer. That's a bit more humane to kill them because there's no way they're not going to be in pain or potentially maimed. You could potentially gather them and raise them. You could donate them to your local school and maybe have the kids in the school raise mantids because they're really fun to raise, and you could feed them with feeder crickets at the pet store. So if you know anybody who's like in doing sort of summer camps or school or any sort of educational thing, maybe give them some mantids because the fact is they're concentrated in this meadow, but there's places in Connecticut where it's really hard to find mantids. So maybe they just need to be brought somewhere else and not just concentrated in this one meadow. So I would say relocate them, raise them in captivity, or if you're really desperate, put them in the freezer to kill them. But as far as the egg cases go, the egg cases are kind of like, it's almost like a insulating foam. The eggs are encased in like this insulating foam. So these are a lot easier to dispose of. You could literally gather them up because they're stuck to the branches. So in the fall, you could see them very easily on the dead stalks of vegetation. You can gather the stalks up, throw them in a bonfire, or you could basically cut them all up, put them in a container and freeze them. And freezing will usually kill anything insect related if you keep them in the freezer long enough. So they're a lot easier to dispose of because the Uthika, the egg masses, are only about like an inch or two across. So they're easy to gather. You could gather hundreds of them and dispose of them in any way you want. Far easier than you would the adults.
Thank you.
Yes, someone said I destroy egg masses. The adults can eat hummingbirds. Yep.
Yeah.
They're voracious. I collected a few for the Yale has an arthropods class, so I collected a bunch of mantids for their entomology class for the kids, and I had a few males with a female. And by the next day, the female had already eaten 2 males before I could even get her to the class. So they're very voracious little things.
Interesting. There's a couple of fun questions here. What's your favorite bug?
Oh,
they want to know.
That's a good question. I'm a moth fan. I'd have to say my favorite moth, my favorite moth are probably Catocala, which are those underwing moths I showed you. It's what the Northeastern United States is one of their centers of diversity. So we can get over 50 species in Connecticut of that particular type of moth. And plus my boss at the Peabody, he's one of the world experts in Catocala, so he kind of got me hooked, and now I'm like a big Catocala nut.
That would be Larry Gall, right?
Yeah, Larry Gall.
June, do you want to tell Ray what your favorite bug is?
My favorite bug is a butterfly.
Nice. Do you have a specific type that you like?
No, I didn't. I actually like a bunch of bugs. I also like spiders. Because when you look really up close to them, you can see all the detail, and I think they look really pretty.
Yeah, I'm glad to see people that like spiders, because there's a lot of people afraid of spiders, but spiders are like, I think there's only one species in Connecticut that can hurt you, and that's the black widow. All the other spiders are perfectly harmless, and they're beautiful. Like if you look at them, they're very beautiful creatures. We didn't even study spiders much. We did see a few spiders, crab spiders and some nursery web spiders in the fields, but we didn't do much spider work this summer.
Someone's asking if there's a native mantis in Connecticut.
That's a good question. The two dominant mantids are the Chinese mantis and the European. So there is also another Chinese mantis. It's called, I think, Tenodera angustipennis. It's called the narrow-winged mantis. It's kind of it looks like the Chinese mantis, but it's a little bit skinnier. So there's 3 exotic mantises in Connecticut. There's one species of native mantis, I think it's called Stagomantis. It's called the Carolina mantis is the common name. And I have yet to see it in Connecticut. And that's another thing where I wonder if these native ones are being actually eaten by the Chinese mantids. So there is one native mantis that's supposed to be in the state, but it's quite rare. And if you see it, you're pretty lucky.
Can you speak about the lanternfly and the impact it may have in our region?
The lanternfly, yeah, that's another good, because my family is actually from Southeast Pennsylvania, and that's the hub, the epicenter of the lanternfly scourge. And they are coming. Like I think their first reports from Connecticut were this year in I think Fairfield County, and I think there actually was a report from Milford or something, but I think it might have been associated with greenhouse transport. But they are coming. The adults are large and mobile, and they're really easy to spot. So everybody who's living in the southwestern part of Connecticut needs to keep an eye out for the lanternflies. They're very large and very pretty. They're almost, a lot of people think they're a moth when you see them, because they have these pretty spotted wings with these red hind wings. And but they're very serious pests to plants with soft stems, because as nymphs, they're colonial, so they live in these large groups and they suck the juices out of plants. And then as adults, they're big, so they're potential danger for crops, grape crops and fruit crops, because their piercing mouth parts will spoil the fruit. So both the juveniles of the species and the adults are pretty devastating pests. So if you do see a lanternfly, contact the local authority, like contact DEEP, which is the Department of Environmental Protection in Connecticut. If you see anything like a lanternfly, make sure you notify the authorities very quickly.
Good, very good to know. If you were to elect a native state insect, what would you nominate?
I was thinking that because I was actually looking to see if Connecticut had a state butterfly, and we don't have an official state butterfly, which is a little bit strange to me, because I mean, most people would take something like usually it's the tiger swallowtail or the monarch. They do like a safe pick. I would pick something like the Baltimore checker spot or something like that. That was a much more beautiful, unique species, because the Baltimore checker spot is found mostly in the northeastern Appalachian region. So I think it's a little bit prettier and more unique example for a state butterfly. A better state insect, I would have to think more hard about that, because I'd have to think what's a really a native species that's like really, because if you think of an insect off the top of your head, you're going to think that's really common. You're going to think of something that's not native. You're going to think of the lady beetle or the brown stink bug or the Chinese mantis. But there's so many potential choices. I don't have an answer right off the top of my head for that.
Yeah, it was just a fun question, you know. And someone's asking now too, what I guess the iNatural site ID can the moths records be found under? Steve is asking that.
What INAT ID can the moth? Oh, I guess you wanted to know if there's a repository where this is all the moth records are, that the stuff I showed from Fresh Meadows could be found.
Yeah. And Steve, feel free to unmute and speak if that's not correct or you want to add more to the question.
Well, yeah, I've done a lot of moth surveying, mostly on Long Island. Also, I used to do some stuff in Westchester, and not really an official survey, but I did some black lighting in Litchfield County a number of years ago. So, you know, I might have some interesting perspective on some of the records, like the English name for one of the moths you showed was an eldershoot borer moth, which I do have a Connecticut record of from about 10 years ago.
Yeah, that was the first one that, Acatodes zea, I think it was called. That was one of the first one I found in Connecticut was at this particular site. But again, like I said, it's not, there's probably other records other people have found in museums. I'm more speaking of firsts for iNaturalist, which is not, there could be a lot more records for Connecticut that aren't actually, I'm not aware of, because I did do like searches of the USNM and the American Museum of Natural History in the Peabody for records of all these species. So I'm going by just stuff that's normally found on iNaturalist. It's more common that we got the first reported records for the state.
Yeah, I suspect that's pretty uncommon, or it just doesn't come to light, because I've only seen 2 of those in 20 years. So that's a good one.
And I've done significant lighting up in Canaan, in that area of Connecticut, which is a beautiful area. Like, that's probably the hot spot of diversity in the state. But I've never come across that particular species, so it was definitely notable to me.
Thanks, Steve. Karen's asking, how about a walking stick for a native insect?
That's a pretty good, there's two species in Connecticut. They're both native, so that would potentially be, they're not the commonest thing. So usually when you pick a state insect, you want to pick something that everybody knows. But I think most people, when they see a walking stick, they know a walking stick. They don't have to know the particular species of walking stick. But that's a good idea.
Yeah. So there's some other comments in the chat box. Would anybody else have any other questions or comments? If you can feel free to unmute yourself. Oh, June, go ahead, June. Unmute yourself, hon.
Once I
June,
heard,
just a second. Go ahead, June.
once I heard this thing where somebody gave a bunch of caterpillars a whiff of a smell, and then they gave them a shock a few times, and then once there were like hundreds of them. Once all of them were butterflies, they did the same thing. This time they flew away. They did this test to see if butterflies, to see if caterpillars still had their memories from when they were butterflies, because once they opened a cocoon and inside there was just like a white jelly thing. And they noticed that it still saves its memories while it's still like that white ball of jelly.
Wow. Oh, so when it's metamorphizing in the chrysalis, it turns into the jelly. That's cool.
Yeah, cool. Thanks, June. Jim, I'm sorry, you were
Yeah.
trying to ask a question. Yeah.
With this wide diversity of insects, is there a hierarchy in terms of a dominant species in the meadow?
Ooh, that's a really good question. There were some times when we went, like when we first showed up, those exomala beetles were super common, but they pretty much petered out. By the time we went back in August, they were gone. And they only flew really early. There were several, I would actually have to look at, actually I probably can do that. I'm just gonna go and see the iNaturalist, because I could go to that and sort by location. Get my most common species, so 260. There's a few that we got significantly more than others, depending on the time we were there. We got 9 Catocala ilias, 5 Ultronias, a bunch of Amphipyra, which is a copper underwing. Those procaroids, those large maple spanworms were very common. The juniper geometers were very common, and those idia moths were also fairly common over the course of the summer. So there's a bunch of species that are quite common. And if you look at the sheet, like into the small, if you go to the smaller level, to the microscopic, almost microscopic level, there's a lot of small gnats and midges that were fairly common every time. I think actually Microcrambus elegans, which is a micro moth, was probably the most common moth at the sheets when we were there.
Cool. Awesome. Anyone else have anything? We're almost at the top of the hour here.
I saw a question about the iNaturalist thing, and
Yeah.
that's a good thing to mention, because I don't think I mentioned what my thing is on iNaturalist. My name on iNaturalist is Ray, just my name, Ray. So you'll see me on there. If you look any moths basically in Connecticut, you'll see my name on there.
Great.
And Steve's is Steve 432 King or something, and then Danny's is Colivia. So the 3 of us are all on iNaturalist. There is no project for Fresh Meadows. I think if we continue doing this, if we continue doing these surveys next year, I might create that project, and that would make it a lot easier for people to find this data. But if you go on iNaturalist and search for Connecticut, you can search for basically the town of Cheshire, and just sort by everything found in that town, and you'll see a lot of our records pop up just by searching for Cheshire.
And I just want to let people know too that Cheshire Land Trust has another presentation with Doug Tallamy is going to present on Wednesday, November 11th at 6:30. So you can go to CheshireLandTrust.org to register for that and to find out more information about us. Thank you so much, everybody.